My name is Richard Logan, and my presentation today will be on adhesion junctions.
So what are adherent junctions? They are cell-to-cell adhesion complexes that are continuously assembled and disassembled, allowing cells within a tissue to respond to forces, biochemical signals, and structural changes in their microenvironment. There are anchoring junctions, binding bundles of actin filaments between adjacent cells via the protein cadherin, which are supported by other proteins called catenins, mainly alpha and beta catenin, which you'll learn later about in the PowerPoint. And they are found mostly in tissues under severe mechanical stress such as the heart, muscle, and epidermis.
Adherence junction is an adhesion belt made of plaque that surrounds a cell. Transmembrane glycoproteins, in this case we're going to call cadherins, they join the cells together. This is good for keeping epithelial surfaces from slipping, sliding out of position. Let's take a look at what this kind of looks like. Now, using the arrows for an example, what we're seeing here mainly is this going to keep one thing from sliding down and the other from sliding up. So kind of like a shear force, if you will. So where this black arrow is pointed to, this is an adhesion belt that runs around the cell. And where the second black arrow is pointed to, this green little cadherons stick out from this plaque belt and they kind of catch on each other and they keep this thing from sliding again. Now back to the first arrow's example. This idea is where we're keeping sliding motion from occurring. So these things won't slide past each other. Here's another close-up view of what we're seeing here with the cadherons interacting with each other in the inner membrane space. And he's circling.
The adherence junction is an essential feature in mammals. It provides a protection to our skin, a la epidermis. On top of the ability to contract, the epithelial and endothelial tissue makes this junction an important factor when it comes to embryonic development. It gives rise to the structure of neural tube and lens of the eye. The adherence junction also regulates the vascular permeability, of which their impaired function can lead to several pathological conditions. Examples of these pathological conditions are inflammation, sepsis, ischemia, and diabetes. Listeria monocytogenes is an intracellular bacterium and is a causative agent of listeriosis. It is one of the most virulent foodborne pathogens, with 20 to 30% of clinical infections resulting in death. It causes invasive disease by crossing the intestinal epithelial barrier. Biochemical studies have shown that both tight junction proteins and adherent junction proteins are degraded in apoptotic cells, which may facilitate their detachment from neighboring cells. By detaching, these junctions are no longer able to function and encounter apoptosis.
So the study I've chosen was on the role of adherent junctions in the developing neocortex. The disproportional enlargement of the neocortex through evolution has been instrumental in the success of vertebrates, in particular us mammals. The neocortex is a multi-layered sheet of neurons generated from a simple proliferative neuroepithelium through a myriad of mechanisms with substantial evolutionary conservation. This developing neuroepithelium is populated by progenitors that can generate additional progenitors as well as post-mitotic neurons. Subtle alterations in the production of progenitors versus differentiated cells during development can result in dramatic differences in neocortical size. So the experiment examined how cadherin adhesion proteins, in particular A-catenin and N-cadherin, which we talked about earlier in this PowerPoint, function in regulating the neuroprogenitor microenvironment, cell proliferation, and differentiation and cortical development. So the strength of the experiment showed that the apical concentration of adhesive and signaling molecules may provide an extra nuclear hub utilized to sustain the proliferator neuroepithelium during development. So due to this finding, it enabled future studies to investigate the rules of these apically concentrated signaling molecules, how they play in maintaining a proliferative microdomain or niche. And it should be fruitful and elucidating, because moreover, understanding which pathways intersect and how and when they intersect will be useful for not only for the field of development neurobiology, but many others like cancer, where alterations in cell-to-cell adhesion impact cell behaviors.
So my question to y'all is, what further research can be done to discover new novelties for adherence junctions?